Welcome to this presentation. We are going to look at fetal development. So by the end of the session, you should be able to know the process of fertilization, the different stages of fetal development, then the different embryonic and fetal structures. So the process of fertilization occurs when the ovum fuses with the sperm cell. Okay, the ovum is released during ovulation. Okay, this usually occurs around the 14th day of the menstrual cycle, and the spermatozoa or the male gametes are released into the female reproductive tract during sex or intercourse, and then they travel to the fallopian tubes where fertilization happens, the place called the ampulla. The sperm cells can remain viable in the female reproductive tract for about 24 to 72 hours. That is 1 to 3 days. We have to recognize the importance of the conducive environment or the conducive the environment conducive for the viability of the sperm cell. And one of the key roles of the mucus that is produced by the cervix, one we call the alkaline cervical mucus, is to provide that conducive environment. It is alkaline, that means that it has a pH of above 7, and this actually helps in neutralizing the acidic vaginal environment, and therefore this facilitates the survival and transportation of the sperm cells. Okay, so around the ovulation time, that is when this alkaline cervical mucus is actually produced. It also contains nutrients that can support sperm viability and function. So fertilization basically occurs when the male nucleus, the female nucleus fuse. Okay, this happens after a series of reactions that chemical in nature, resulting into a release of enzymes that alter the surface or the zona pellucida, which is the membranous surface of the ovum, such that no further sperm cell can enter. Okay, the fertilizer cell. So the sperm cell has to penetrate a number of membranes, the radiator cells, okay, the radiator cells, and then the zona pellucida, then the vitelline membrane. Okay, its penetration is aided by a reaction which we call the acrosomal reaction, and this is depicted in the picture that we have. The first step is that when the sperm cell makes contact with the egg, which is the ovum, you will have lytic enzymes released from the acrosome. And these lytic enzymes react with the zona pellucida, which is the outermost layer that you can see there. And then The next step is that the acrosome also reacts with the perivitaline space, okay? And then finally, where we will have the plasma membrane of the sperm cell and the egg fusing, okay? And then now the sperm nucleus can enter the egg, okay? Then you have parts of course of the that is the tail breaking off, and then the neck, okay? You also have cortical granules fused with the plasma membrane, which render the vitelline membrane impermeable to the sperm cells or to other sperm cells. Okay, so that is the fertilization for you. After fertilization, just a minute, I think that slide came in. So we have another very important step or process that we call capacitation. Capacitation is the process that prepares the sperm for fertilizing the egg, and it starts at the moment when the sperm enters the female genital tract, the uterus. So you have a lot of, you have biochemical processes, biochemical and biophysical processes or changes that occur in the sperm membrane, okay? We have ions being involved, we have proteins being involved, and cholesterol efflux being involved. These changes, these biochemical changes, are very important because they prepare the sperm cell to undergo a chromosomal reaction, which we have earlier on explained. Again, a review of the chromosomal reaction. You need the sperm cell to penetrate the different layers, the coronal radiator cells, the zona pellucida, and then the vitelline or the perivitaline membrane. Okay, so you need the sperm cell to penetrate through that, and the chromosomal reaction enables that, where you have hydrolytic enzyme being released, and these enable the penetration to happen. So when we talk about the stages of development of the fetus, we can classify the stages into three. Okay, the pre-embryonic stage, the pre-embryonic stage, which lasts between 1 to 2 weeks, and then the embryonic period, which is called organogenesis, lasts from the 3rd week to the 8th week, and then the fetal period, which lasts from the 9th week to term. It's about 38 weeks, 38 to 40 weeks, okay? So the next 3 steps or the description, after fertilization, you'll need the zygote to be propelled to the uterus for implantation. And before implantation, what really happens? Before implantation, you'll have the zygote multiplying, okay? That is by cell division, increasing in cell number, and become what we call the blastomere. Then the blastomere will have a fluid, will be filled with fluid to form the blastocyst. So you have a series of cell divisions that have mitotic cell divisions that have happened to come to that. And then this now is moved to the uterus. Okay, so you have these divisions happening as the movement to the uterus happens. And then before the implantation, you need to have that zona pellucida, which surrounds this zygote, to disappear before the implantation really happens. So that stage is basically what we call the pre-implantation stage. So let's really look at the exact processes that really happen. When the zygote has been formed, it will undergo a series of cell divisions or mitotic cell divisions, still within the zona pellucida. Okay, the zona pellucida is what covers this the zygote at this moment. Okay, and these cell divisions can start within the first 24 hours after fertilization. This type of cell divisions are called cleavage. Cleavage. Okay, the cells increase in number, but the size does not really change, and it can occur for up to 6 to 7 days. So these divisions give us what we call the blastomeres. Okay, and these divisions happen as the zygote is being moved the uterus for implantation. That movement is by 2 processes: the peristaltic movement of the fallopian tubes and the ciliary motion. Okay, and by the time the blastomere or the zygote reaches the uterus, it is now solid mass of cells which are uniform. And by this time, the cell divisions that have happened are between 15 to 50. And this solid mass of uniform cells is what we call the morula. So how these mitotic divisions happen can be subdivided into 3. Initially, you have 2 cells that have fused, and then these 2 cells now undergo multiple divisions to form blastomeres, and then finally forming what we call the morula. The morula is, can consist of 16 cells by the 4th day after fertilization. And by the time we have 16 cells, we have had about 4 divisions, 4 cell divisions, and still covered by the zona pellucida, which we said should disappear at the process during implantation. Okay, so this morula now consists of 2 layers of cells: the inner cell layer and then the outer cell layer. The outer cell layer is the cells that form it are the trophoblasts, and then the inner cell layer is what we call the embryo proper, or we can call it the embryoblast. Okay, embryo proper or the embryoblast, or we can also call it the inner cell mass. Okay, now we have talked about the pellucida. It has to disappear before implantation happens, but one of the roles of the zona pellucida is that it allows the zygote to actually implant in the right site. Otherwise, you'll have the zygote implanting in the wrong site, causing what we call ectopic pregnancy. Okay, so we also said that you will have more cell divisions happening to form what you call a blastocyst. And the blastocyst can consist of about 32 to 64 cells. That means this happens after several other cell divisions. And then you'll have fluid that pass from the uterine cavity to enter into this morula to form the blastocyst. Okay, so that fluid in the blastocyst is the blastocell. Okay, and therefore it is this blastocyst that will actually attach the endometrium to, that is basically the process of implantation. Okay, the process of implantation involves the blastocyst attaching itself to the endometrium with the help of the outer cell layer, which we said are the trophoblasts. So the trophoblasts play an important role in invading the endometrium of the uterus for fertilization. So implantation can happen between the 6th to the 12th day after fertilization. During implantation, a woman might see some spotting and may mistake this spotting to be her normal menstrual blood, yet it is actually implantation spotting. Okay, after implantation, that is when we now have the embryo. Okay, so we said that we have a blastocyst which has a fluid-filled cavity, and then we have the embryo proper, or the inner cell mass, or the embryoblast, which we can also call the inner cell mass. And then the outer layer of cells are the ones we call the trophoblasts. So we have the inner cell mass moving towards one end, towards one pole, with that pole and call the polar trophoblasts. Okay, so we will have the blastocyst having different cells arranged differently, and also giving rise to different structures. And the focus here is that I want you to focus on the epiblasts, which are the blue cells, and then the hypoblasts, which are basically the yellow cells that you see there, basically around the inner cell mass. Okay, so the inner cell mass is the one we said that the one we said is called the embryoblast. Okay, called it the embryoblast earlier on. It's the one which gives rise to the 3 germ layers: the ectoderm, mesoderm, and the endoderm. It is very important to understand these layers, because these layers are the ones that give rise to different tissues and organs of the embryo. So the epiblasts, the ones that face the trophoblasts, they lie on the outer shell of the blastocyst. Okay, whereas the hypoblast is the layer of cells that face the blastocell, which is the fluid that the fluid-filled cavity inside the blastocell. Okay, the epiblasts give rise to the 3 germinal layers that we talked about, that give rise to different organs and tissues of the body: the ectoderm, mesoderm, and then the endoderm. The hypoblasts give rise to the yolk sac and the chorion, which are structures that actually support the embryo. The epiblasts are pluripotent. This is to say that they can differentiate into different types of cells in the body. Okay, that's a continuation. On the other hand, the hypoblasts are not pluripotent. Okay, they cannot form other types of cells. but they play an important role in the formation of the primitive strake, which is a linear band of cells that mark the beginning of gastrulation. Gastrulation is a stage in the embryonic development where we have that two-layered structure, the epiblast and the hypoblast, that we say that give rise to the ectoderm, mesoderm, and the endoderm. So the germ layers that we have been talking about are very important because we have organs arising from them. So we need to know which organs arise from which germ layer or germ disk. So the ectoderm gives us the central nervous system, the sensory organs, the skin, the hair, the nails, the tooth enamel. Some endocrine glands, such as the anterior pituitary and the adrenal medulla, also come from the ectoderm. The mesoderm gives rise to the connective tissues. If you, I can use that. Muscle. We have bones, cartilage, we have muscles, the blood, lymphatic system, and then the cardiovascular system, urinary system, and most of the reproductive system. It also gives rise to some of the endocrine glands, such as the adrenal cortex and the gonads. The endoderm gives rise to the gut of the embryo, and therefore forms the digestive system, the respiratory system, the liver, the pancreas, thyroid, parathyroid, and then thymus. Some of the endocrine glands also come from the endoderm, such as the posterior pituitary. Trophoblasts, we have talked about them. We say that they are the outer covering, the cells that form the outer layer of the blastocyst. Okay, and they play two important roles. One, providing nutrition to the growing embryo, and then two is that they facilitate the implantation. They are the ones that invade the uterine wall for implantation to happen. There are two types of cells. The trophoblasts consist of two types of cells: the villous cytotrophoblasts and then the extravillous cytotrophoblasts. Okay, so the villous cytotrophoblasts proliferate and fuse to form the syncytiotrophoblasts, which cover the surface of the placenta and are in direct contact with the maternal blood. The extravillous cytotrophoblasts cells migrate out of the placenta and invade the maternal tissues, such as the endometrium, the myometrium, and the spiral arteries. So the process of implantation said that the trophoblasts are responsible, but this process has to be regulated. Okay, and this regulation is by molecular and cellular interactions between the trophoblasts and the maternal microenvironment, which include factors, or what we call autocrine and paracrine factors. Okay, the trophoblasts also play an important role in ensuring that the maternal immune system does not invade the implanting embryo. Okay, so it makes the endometrium receptive to the implanting embryo. So basically, implantation can happen between the 6th to the 12th day, and it happens if the covering of the blastocyst actually disappears. That is the zona pellucida. And then the endometrium has to change into the deciduum. Okay, the endometrium has to change into the deciduum for implantation to really happen. And then you will have the inner cell mass, the formation of the body stop connecting the inner cell mass to the trophoblast, and then, of course, the changes in the endometrium facilitate the implantation to take place. And then you also have formation of the bilaminar disc, which we earlier on talked about, the epiblast and then the hypoblast. It is the epiblast and the hypoblast that will form the germinal layers. And the germinal layers are the ectoderm, mesoderm, then the endoderm. And these are the three, the ones that give rise to the bone structures. That's what we have talked about. So we said that the endometrium has to change. Okay, so the modification of the endometrium, the mucosal lining of the uterus has to change during pregnancy, and we call it the decidual. Okay, so for these changes or decidualization to happen, we have changes in structure and function of the endometrial stromal cells, of connecting tissue cells and their interaction with other cells, such as the immune cells and blood vessels. Okay? This process happens under the influence of hormones, such as the human chorionic hormone, HCG. Okay? The human chorionic gonadotrophic hormone. This is the hormone that we actually we are testing for humans as one of the preliminary tests if you're testing for pregnancy in humans. So the decidua is very important because it prevents the attack by immune cells from the maternal site from attacking the implanting embryo. Okay, it also provides nourishment and plays an important role in formation of the placenta. Okay, the decidua has three distinct parts: decidua basalis, decidua capsularis, and then decidua parietalis. Okay, the part that covers the embryo is the decidua basalis, and then the next part is the decidua capsularis. Okay, it overlies the embryo and covers it from the uterine cavity. Then the last one is the decidua periostealis. Is the rest of the decidual tissue that covers the uterus. Okay, so decidualization is the adaptation of the uterus to enable implantation and maintain pregnancy. Hormones such as HCG that I've talked about are very important, as well as progesterone, also very important. This process can be impaired by factors such as genetic factors, environmental factors, and this can result into complications or loss of pregnancy. So we look at the chorionic villi and the placenta. Around the 11th to the 12th day, you have chorionic villi being formed. Okay, the chorionic villi are just projections of the chorion that provide maximum contact with maternal blood for nutrients and gaseous exchange. So those projections that you see, the ones we are calling the chorionic villi, they are also very important to the roles of the placenta. The chorionic villi develop from the trophoblastic cells, the trophoblasts of the blastocysts. Remember, the trophoblasts are the outer cells of the blastocysts. And so that means that the blastocysts play an important role in implantation and also in formation of the chorionic villi, therefore the placenta. So the development of the chorionic villi can be put into three: primary, secondary, and then the tertiary. Okay, so you will have the primary lacking the blood vessels, and then the secondary now becoming large and also branched, and having mesodermal cells, the blastocytes, blast cells. Okay, and this appears around the 16th to the 21st day of gestation. And then the already developed villi are the ones that we call the tertiary villi. And these ones contain blood vessels. Therefore, we say they are vascularized. And these blood vessels come from the umbilical cord and appear between the 17th to day 22 of gestation. So the placenta is an important structure where you have the embryo deriving its nutrients from it. Nutrients, oxygen, antibodies, hormones, all this diffuse through the placenta. So the placenta has the fetal part and then the maternal part. The maternal part is called the sudor basalis, and then the fetal part is the chorionic frondosum. Okay, so the frondosum is the part of the chorion that develops the numerous chorionic villi, which actually invade and destroy the uterine deciduum and absorb nutrients, what you see here. Okay, the chorionic frondosum. Okay, so we said that the chorionic villi form from the trophoblasts and play an important role in formation of placenta. And these syncytial trophoblast cells actually produce hormones such as HCG and also play an important role in protection of the fetus. The cytotrophoblasts can differentiate into extravillous trophoblasts, which also invade the uterine wall deeper, okay, deeper into the uterine wall, and also help in formation of the maternal spiral arteries to increase blood flow through the placenta. We also talk about the placenta as a barrier. That means it prevents the diffusion of toxic material or chemicals or substances to the fetus. So this barrier is formed by the syncytial trophoblasts, cytotrophoblasts, connective tissue, and endothelium of the fetal. Okay. So the placenta also produces hormones. We already looked at the HCG, which helps in sustaining the corpus luteum. That is very important in producing estrogen and progesterone, which are vital in supporting pregnancy. Estrogen, produced by the syncytial cells, contribute to uterine growth and development of mammary glands in preparation for lactation, whereas progesterone maintains the uterine lining during pregnancy and reduces uterine contractions to prevent premature labor. Another hormone, also called human placental lactogen, promotes development of mammary glands, lactation in the mother. Okay, so the chorionic villi are very important in enabling exchange of nutrients and also gases such as oxygen between the maternal blood and the fetus. Okay, so we have what we call the intervillous space. This is the space between the chorionic villi and the decidual salix. This is where this exchange happens. Okay. And then we have the anchoring villi. These are the villi that actually are attached to the basalis. So we have those that are not attached, therefore there is space. And then we have those that are attached, and provide stability to the placenta. The amnion is the membrane that basically covers the embryo. If you can see here, okay, that line is the amnion. It covers the embryo, therefore provides protection. It originates from the ectoderm, the mesoderm. Okay, it's one of the extra embryonic membranes that support the embryo alongside the chorion, yolk sac, and the long pulse. The amniotic fluid is the fluid that is slightly yellowish in the amniotic cavity. It ranges between 600. It can be 800 milliliters at 34 weeks of gestation. It can reduce to about 600 milliliters 40 weeks of gestation. This therefore means that it tends to reduce towards time. Okay. So the functions of the amniotic fluid: it protects the fetus, acts as a shock absorber. If there's external trauma, protects against infection, prevents the umbilical cord from being compressed. If the umbilical cord is compressed, then you will have blood supply and oxygen being stopped from reaching the fetus. It facilitates movement, keeps the temperature around the fetus normal, and then also it regulates fetal growth by providing nutrients, hormones, and growth factors. If you have little amniotic fluid, then you have oligohydramnios. So amniotic fluid of less than 500 at term is oligohydramnios. Polyhydramnios is when you have more than 2,000 milliliters of urine, amniotic fluid at term. And then the fetus is connected to the placenta through the umbilical cord. Okay. The umbilical cord has two arteries, then one vein. These play an important role in transportation of nutrients, oxygen, hormones, and other growth factors, including waste products and also antibodies. The arteries carry blood away from the fetus to the placenta, whereas the veins carry blood towards the fetus from the placenta. We can actually say that the pregnancy can be divided into 3 trimesters. The first trimester happening between 0 to 12 week, then the second trimester happening between the 12th to the 25th week, and then the third trimester happening between 27th to 38th week, which is basically time. So along this, during this gestation period, you will have a number of things happening, growth and development of different structures. And in these slides, we highlight which structures develop when and what are the things that you expect to have developed at what period. So I'll end this presentation here. Thank you for listening.